frozen mummy is offering new insights into human history and geography.
The Iceman, as he's known, has been the subject of scientific study since he was discovered in 1991.
When scientists first uncovered the mummified body of Otzi high in the Alps, the world was immediately captivated. Frozen for more than 5,000 years, his preserved form offered a rare and intimate glimpse into the life of a Copper Age man. Everything about him, from the flint tools and copper axe he carried to the tattoos etched into his skin, was a revelation. Yet for decades, his true genetic identity remained a puzzle. Advances in DNA sequencing eventually changed that, and when scientists finally decoded his genetic makeup in unprecedented detail, what they discovered shifted long-held assumptions about him, his origins, and the world he lived in. Before we start, smash the like and subscribe buttons for more updates. The unraveling of Otzi's DNA was not just a matter of curiosity, it was a technical feat, requiring careful handling of degraded genetic material preserved in ice for millennia. Early studies had provided a fragmentary picture suggesting general links to modern European populations, but newer high-throughput sequencing technologies allowed researchers to read his genome with a clarity that had been impossible before. The moment this was achieved, a new narrative began to take shape, one that surprised scientists and added new depth to our understanding of Europe's prehistoric past. One of the first startling revelations concerned Otzi's ancestry. For years, many had assumed that as a man of the Alps, his genetic makeup would align closely with modern Central Europeans. Instead, his DNA indicated a far stronger affinity with early Neolithic farmers who had migrated into Europe from the Near East thousands of years before his birth. These agricultural communities carried their traditions of farming and herding across the continent, fundamentally reshaping prehistoric Europe. What astonished researchers was just how little genetic mixing Ötzi appeared to have had with the hunter-gatherer populations who had once dominated the region. His genome was strikingly unmixed, suggesting that his community was part of an isolated population that retained a distinct identity, even as waves of migration swept across Europe. Another discovery challenged assumptions about his appearance. Reconstructions over the years had often depicted Ötzi with light skin and lighter features, partly based on cultural expectations of ancient Europeans. But the decoded DNA painted a very different picture. Genetic markers indicated that Ötzi had darker skin than most modern Europeans, likely closer to the pigmentation of people living around the Mediterranean today. His eyes were brown rather than blue, and his hair was not the lighter shade often imagined, but dark and potentially thinning even during his lifetime. This darker complexion surprised many because it disrupted ingrained notions of how early Europeans must have looked. It also highlighted how intertwined skin pigmentation is with the deep history of human migrations, adaptation, and environmental pressures. Health was another window that the DNA sequencing opened. Otzi's genetic profile revealed a predisposition to several conditions that continue to affect people today. Most strikingly, he carried genetic markers associated with heart disease. This detail brought new significance to what forensic examinations had already suggested, that his arteries were unusually hardened for a man of his age. It became clear that even in a world without modern processed foods, sedentary lifestyles, or industrial pollution, genetic inheritance could predispose someone to cardiovascular risks. This raised fascinating questions about how prehistoric humans might have contended with health challenges shaped as much by genetics as by environment. His genetic blueprint also confirmed that he was lactose intolerant. This was an important piece of the puzzle, as it reflected a critical stage in human adaptation. While dairy consumption became more widespread in Europe after the spread of farming, the genetic mutation allowing adults to digest lactose had not yet become common in his community. This meant that even as cattle were domesticated and milk became available, people like Otzi would not have been able to process it. The detail underscored how cultural practices such as dairy farming and genetic evolution do not always align perfectly in time, but instead overlap and slowly shape each other. On a broader scale, decoding Otzi's DNA offered a window into the movement of peoples and the persistence of lineages across deep time. His ancestry traced a path back to Anatolian farmers who had spread into Europe during the Neolithic, carrying with them new technologies and ways of life. The fact that Ötzi's genetic makeup remains so heavily tied to this ancestry with little evidence of admixture from steppe populations who would later dominate European genetics, suggested that he lived in a region or community that remained relatively untouched by the great demographic shifts of his era. This painted a picture of an isolated alpine population, clinging to traditions and genetic continuity even as the continent beyond underwent waves of change. Beyond Ancestry, his genome contained evidence of parasites and pathogens. Traces of Helicobacter pylori, a bacterium still notorious today for causing stomach ulcers, were detected. The presence of such pathogens in his system offered a vivid reminder that humans have lived in an unbroken struggle with microbial adversaries for millennia. Far from being diseases of modernity, many of the ailments we still wrestle with were present in deep prehistory, shaping human resilience and survival strategies. For scientists, this opened opportunities to track the evolutionary history of pathogens alongside the humans they afflicted. The surprise of Otzi's DNA went beyond the specifics of his complexion or medical vulnerabilities. What resonated most was how his genetic story recast the narrative of early Europe. Instead of being a product of broad mixing and movement, he represented continuity, a pocket of the past preserved within a population that remained remarkably unmixed. In this sense, Ötzi was not just an individual, but a representative of an ancient community that had held on to its identity against the tide of change. The discovery underscored the importance of local micro-histories in understanding human evolution, reminding us that not all populations were swept up in the same movements at the same times. These revelations also illuminated the complex relationship between culture and biology. Here was a man living at a time when copper smelting, farming, and complex social structures were transforming Europe. Yet his genetic profile tied him directly to the very first wave of farmers who had entered the continent thousands of years earlier. He was both a participant in ongoing cultural revolutions and a genetic time capsule of the past. The juxtaposition was striking. It demonstrated that while human technology and culture evolve rapidly, genetic adaptation often follows at a slower, more uneven pace. Equally compelling was how the decoding of Ötzi's DNA challenged modern assumptions. The revelation of his darker skin tone reminded many of the dangers of projecting contemporary ideas backward into the past. It showed that Europe's prehistoric peoples were far more diverse in appearance than once thought, and that the paleness commonly associated with Europeans today developed later and under specific selective pressures. This realization contributed to a broader rethinking of how scientists and the public imagine ancient populations, and underscored the fluid, ever-changing nature of human identity. From a methodological standpoint, the achievement demonstrated how far science has advanced. A few decades ago, the idea of reconstructing the full genome of a 5,000-year-old mummy would have seemed fanciful. Now, scientists were not only decoding his DNA, but identifying predispositions to disease, reconstructing physical traits, and tracing migration patterns with remarkable precision. This leap in capability underscored the power of genetic science not only to revisit the past, but to connect it directly to the present. The implications of this work ripple outward. By comparing Ötzi's genome to those of modern populations, scientists could trace lines of continuity and divergence, piecing together the story of how ancient migrations shape today's genetic landscape. For example, his strong link to early Anatolian farmers demonstrated how profound the Neolithic Revolution had been in reshaping Europe's population structure. At the same time, the absence of significant steppe ancestry in his genome highlighted the uneven spread of later migrations, reminding us that history is always more complex than broad generalizations suggest. The decoding of his DNA also deepened the personal dimension of Otzi. Before, he was a figure frozen in time, remarkable but distant. Now, genetic science had revealed details about his health, his appearance, and his ancestry that made him feel more tangible. He was no longer just the Iceman, but a man with dark skin, brown eyes, thinning hair, and a body carrying silent genetic risks that would resonate with many modern humans. These details humanized him, allowing us to see him not only as an archaeological marvel, but as a person who once walked the mountains, hunted, and lived with his community. Such discoveries inevitably stirred imagination about his world. What did it mean for a man with genetic risk factors for heart disease to live in a demanding alpine environment? How did lactose intolerance shape the diets and cultural practices of his community? How did isolation from broader migratory waves influence their traditions and identity? Each answer opened new avenues of inquiry, painting an ever richer picture of life in the Copper Age and reminding us of the profound continuity between their world and ours. The decoding of Otzi's DNA did not just expand knowledge of one individual, it reframed how we understand prehistoric Europe. It demonstrated that scientific progress can overturn entrenched assumptions, that the past is always more surprising than imagined, and that each advance in technology can peel back another layer of history. For a man who died more than 5,000 years ago, Ötzi continues to speak with a voice that grows clearer with every discovery, carrying across the ages a message about who we are and where we come from. When the genome of Otzi was finally decoded in full depth, the accomplishment was more than a triumph of technology. It was a portal to the lives, choices, and environments of entire ancient communities. His frozen body had already told researchers stories through artifacts, tools, and clothing, but the genetic record carried something even more enduring. It was the biological memory of generations before him, preserved in every sequence of his DNA. In extending the exploration, scientists began to use these findings not only to reconstruct who he was, but also to situate him within the wider drama of human evolution, the spread of farming societies, and the enduring adaptation of humans to extreme environments. One of the most fascinating dimensions that unfolded from the study of his DNA was the ability to infer the mobility of ancient peoples. The genetic signatures tied him to Anatolian farmer ancestry, but this connection also suggested larger migratory networks that stretched across Europe thousands of years before he was born. These networks were not random drifts of people but structured movements, where agriculture, animal domestication, and village life spread systematically into regions once dominated by nomadic hunter-gatherers. Ötzi's genetic background revealed that, at least in some alpine pockets, those first farmers did not simply vanish into a tide of later arrivals. They endured, conserving their biological and cultural heritage. This was an important realization because it demonstrated that prehistory was not a story of populations sweeping away one another in successive waves, but often one of coexistence, isolation, and preservation in unexpected corners of the continent. The decoded DNA also became a mirror through which scientists studied the interplay between genes and environment in extreme living conditions. The Alps, while beautiful, posed profound challenges for survival. Thin air, cold temperatures, rugged landscapes, and limited agricultural zones made life demanding. The genetic profile of Otzi allowed researchers to search for adaptations that may have helped his community thrive in such conditions. While he showed no evidence of traits like those seen in later high-altitude populations, his genome reflected a careful balance of inherited traits and cultural innovations. For example, genetic vulnerability to lactose intolerance would have forced his group to derive nutrition through alternative methods, such as fermented foods, meat preservation, and cereal cultivation adapted to mountainous terrain. Their survival was the result of this dynamic interdependence between inherited biology and the ingenuity of culture. Tracing his genome also illuminated the deep structure of social relationships during the Copper Age. Every human carries clues not just of ancestry, but of kinship networks that shaped their communities. Scientists could compare Otzi's DNA to ancient and modern populations, highlighting who he was most closely aligned with and where his group may have drawn their partners. The relative genetic isolation suggested small, tight-knit groups with limited external exchange, especially compared to later Bronze Age societies characterized by greater mobility. This level of isolation offered a fascinating glimpse into how geography, such as the isolation of alpine valleys, could foster unique communities that stood apart even while Europe grew more interconnected. Such insights allowed archaeologists to imagine not just how he lived, but the scale of his community, the webs of kinship that sustained it, and the barriers that defined it. The power of his genome lay not only in what it said about him, but in what it revealed about disease ecology thousands of years ago. The traces of Helicobacter pylori in his gut were one such example. This bacterium is still common today, but its detection in Otzi's body allowed scientists to reconstruct the evolutionary path of the pathogen, showing how it migrated alongside humans and adapted over time. Through him, we glimpse an ancient epidemiological story, one in which humans and microbes coexisted in a delicate, sometimes devastating dance. Beyond this specific pathogen, the broader lesson was that every human, even in remote mountain valleys, was enmeshed in microbial ecologies that spanned continents. This interconnection extended the story of his life into the microscopic domain, one as significant for human history as wars, migrations, or inventions. Another revelation born from his DNA was its contribution to understanding the evolution of immunity. Otzi carried genetic markers associated with the immune system that provided clues to how ancient humans fought infections. His particular profile highlighted the delicate balance between robust defense against pathogens and the risk of autoimmunity, a tension still at the heart of medical science today. By comparing his genetic immune markers to those of modern humans, scientists could track how selective pressures over millennia shaped immunity in response to shifting disease landscapes. His genome became part of the larger effort to map how humans adapted biologically to invisible adversaries long before antibiotics or vaccines. Beyond pathogens, his DNA illuminated an important aspect of dietary adaptation. His lactose intolerance, already noted, told one side of the story, but his genome also revealed the broader nutritional strategies of his ancestors. They had not yet developed widespread tolerance for dairy, but their survival was nonetheless guaranteed through agricultural strategies, cereal grains, legumes, and livestock used for meat, hides, and secondary products like cheese, which removed much of the lactose. These findings reframed the Neolithic Revolution as a process not only of cultural innovation, but also of genetic lag and gradual adaptation. The genome of Otzi gave a concrete example of how human culture could leap ahead, creating new resources, while biology struggled to catch up. It also showed how innovation and adaptation overlapped in complex ways, shaping the lived experience of ancient populations. Physical appearance, often underestimated in significance, gained new meaning in light of his genome. The discovery that Ötzi had darker skin than imagined prompted deeper reflection on the evolution of pigmentation in Europe. His complexion was an indicator that light skin had not yet become dominant in many parts of the continent by the time of his death. This in turn suggested that environmental and cultural factors, such as the reliance on cereal-based diets that reduced vitamin D intake, played a later role in selecting for lighter skin. The detail mattered because it revealed how evolutionary processes are layered, contingent, and slow. For centuries, humans carried traits that no longer aligned perfectly with their environments, surviving nonetheless through culture, diet, and resilience. His DNA made it clear that ancient Europeans were not the homogeneous image once painted by earlier scholarship, but a diverse collection of peoples with different adaptations and traits. The sequencing of Otzi's genome also touched on the profound issue of human identity. While we often define ourselves through nations, ethnicities, or cultures, the genetic revelations of the Iceman suggested identities that were far more fluid and unexpected in prehistory. Here was a man genetically tied to ancient Anatolian farmers, physically present in the Alps, and culturally enmeshed in the Copper Age. His identity was a blend of inheritance, geography, and innovation. This blurring of boundaries invited a reconsideration of how humans throughout history have understood themselves, showing that what we think of as fixed identities are instead shifting intersections of biology and culture. Equally fascinating was the way Otzi's DNA connected to environmental archaeology. The preserved body itself had already revealed traces of his last meals and the plants that grew in his environment. Combining these findings with his genome provided a fuller understanding of how genetics and environment interacted. For instance, his inability to digest lactose meant that his diet had to be adapted, and analysis of his gut contents confirmed a reliance on grains and meat rather than milk. This link between genetic predisposition and actual dietary practice showed how human lives were shaped at the intersection of biology and ecology. It also helped reconstruct the strategies that Copper Age communities used to survive, from food storage to seasonal hunting. As more genetic details came to light, scientists also began to explore how Otzi's genome contributed to the reconstruction of ancient population maps. By comparing his DNA with modern and ancient samples, researchers could model the movement of peoples over thousands of years. This showed not just where Otzi's ancestors had come from, but also highlighted regions where related lineages persisted longer than expected. These reconstructions challenged older models of sweeping, uniform migrations, replacing them with a patchwork of overlapping movements, retreats, and isolated pockets. Otzi's genome, in this sense, was a needle that helped thread together the larger tapestry of European prehistory. The decoding of his DNA also raised provocative questions about health and longevity in ancient times. While modern lifestyles are often blamed for cardiovascular disease, Ötzi's genetic predispositions suggested that such conditions were not purely products of industrialization. Instead, they were deeply rooted in human biology, existing long before modern risk factors emerged. This reframing showed that even in worlds without processed food or pollution, human bodies carried vulnerabilities. Such discoveries connected us more directly to our ancestors, emphasizing that their struggles with health were not wholly different from our own. A less expected dimension of his genome was its capacity to reveal evolutionary dead ends, traits, and lineages that persisted for a time but did not ultimately survive into the broader population. Certain genetic markers found in Otzi were rare or absent in later Europeans, suggesting that some branches of humanity ended quietly in alpine valleys, their distinctiveness preserved for a few generations before merging or fading. This dimension added poignancy to his story. He was not only a link to the past, but also a record of paths not taken, reminding us that human history is full of lost possibilities and forgotten identities. The reconstruction of his genetic profile also pushed the boundaries of what it means to study an individual. Unlike kings, generals, or cultural icons preserved in written histories, Otzi was an ordinary man, yet his DNA became as informative as entire libraries. This democratization of history, where even the genetic legacy of one person can illuminate entire epochs, shifted the way scientists and the public view prehistory. Otzi demonstrated that the past is not only written in monuments and artifacts, but in the silent scripts of our own biology. Another remarkable aspect of the genomic work was how it redefined the connection between ancient humans and climate. Living in the Alps during a period of climate fluctuation meant that Otzi's community faced shifting resources, changing weather, and potential competition for arable land. His genetic profile provided a baseline to ask whether communities adapted biologically to these changes or relied more on technological innovations like clothing, housing, and agriculture. The evidence suggested that in his time, cultural adaptation was the dominant strategy, with biology trailing behind. This highlighted a profound truth about humans. We are creatures whose survival has always been driven more by invention than by rapid genetic change. The discovery also set the stage for broader interdisciplinary collaboration. Geneticists, archaeologists, anthropologists, and climatologists all found new avenues of study through Ötzi. His genome became a shared resource, drawing connections between human biology and human culture in ways that had rarely been possible before. Each new revelation about him opened questions in other fields, making him a unifying figure in scientific inquiry. His story showed how deeply interconnected disciplines can become when one extraordinary find bridges the gap between them. The decoding of his DNA reminded us of the sheer unpredictability of discovery. When Otzi was first found, he was hailed as a time capsule of Copper Age life, but few could have imagined that his genome would one day be sequenced in such detail. The surprise was not only in what the DNA revealed, but in the realization that every preserved individual holds untapped potential to rewrite history. As sequencing technologies advance, more ancient remains will undoubtedly yield similar revelations. Yet Otzi will always remain singular, both because of the extraordinary preservation of his body and because of the pioneering role his genome played in opening this new chapter of exploration. The Iceman's DNA is more than a scientific curiosity. It is a reminder that humanity's story is endlessly layered, always open to reinterpretation as new tools emerge. His genome holds the memory of migrations, the shadows of ancient diseases, the resilience of isolated communities, and the intricate dance between culture and biology. By decoding it, scientists not only surprised the world with unexpected truths about his ancestry, appearance, and health, but they also reminded us of the profound connections we share with those who lived and died thousands of years before us. His genetic voice, carried across millennia, continues to enrich our understanding of what it means to be human. Otzi's Decoded DNA not only illuminated the details of his life, but also expanded our understanding of the broader scientific methods required to unlock such ancient secrets. Sequencing DNA that has been frozen for thousands of years is no simple task. Ancient genetic material is fragile, degraded, and easily contaminated. The Iceman offered a rare case where preservation conditions were favorable. But even so, scientists had to develop specialized protocols to ensure the authenticity of the data. This effort was groundbreaking because it pushed the boundaries of paleogenomics, setting new standards for how ancient DNA is extracted, sequenced, and interpreted. The lessons learned from Otzi's genome became templates for future studies of other ancient remains around the world, influencing how entire fields of research approached the challenge of reconstructing human prehistory. The technological side of his DNA sequencing demonstrated the extraordinary advances in genomics over the past few decades. In the early 1990s, when Otzi was first discovered, sequencing an ancient genome in its entirety would have been unimaginable. Scientists at the time struggled even with fragments, often unable to distinguish between authentic ancient DNA and modern contaminants introduced during excavation or study. By the time his genome was fully decoded, however, high-throughput sequencing and improved bioinformatics tools allowed researchers to piece together long stretches of genetic code with remarkable fidelity. This technological triumph underscored the speed at which science can transform our ability to ask new questions about the past. The very act of sequencing Otzi's genome symbolized the transition from archaeology as primarily a material science to one increasingly integrated with molecular biology. The decoding of his genome also sparked philosophical discussions about the meaning of individual lives in the deep past. Before the genomic revolution, much of what we knew about ancient peoples came from group-level analyses, skeletal remains, cultural artifacts, settlement patterns. Otzi changed this dynamic. Here was one man whose life could be reconstructed in intimate detail, not only through the tools and clothes he carried, but also through the genetic instructions written in his body. This personalization of prehistory gave scholars a new kind of intimacy with the distant past. Otzi was no longer an anonymous figure among many. He was an individual with specific traits, vulnerabilities, and ancestry. His story represented the meeting point of collective history and personal biography. The implications of his decoded DNA extended beyond anthropology and archaeology into medicine and modern genetics. By examining ancient genomes, scientists can identify when particular genetic variants first appeared in human populations and how they spread. For example, Otzi's lack of lactose tolerance pinpointed a stage before this adaptation became widespread, offering a timeline for the spread of lactase persistence mutations in Europe. Similarly, his predisposition to heart disease suggested that certain genetic risk factors have been present for millennia, long before modern diets and lifestyles exacerbated them. These findings provided valuable insight into how genetics and environment intersect over long timescales, helping medical researchers understand which health challenges are ancient legacies and which are products of recent societal changes. Otzi's DNA also illuminated how ancient humans interacted with their landscape in ways that transcended survival. The presence of Anatolian farmer ancestry in his genome pointed to his community's agricultural roots, but his location high in the Alps suggested that farming alone was not their sole way of life. Archaeologists had already noted evidence of hunting, gathering, and seasonal movement in his toolkit and diet, and the genetic data complemented this picture. His community was not simply a group of static farmers. They were a dynamic society capable of blending agriculture with alpine adaptations. This highlighted how human communities in prehistory were not defined by single identities, but by fluid strategies that combined multiple survival methods. His DNA indirectly underscored the creativity and flexibility of human cultures when faced with challenging environments. A further dimension emerged when scientists considered the role of isolation in shaping genetic identity. Populations that remain isolated tend to conserve older genetic signatures, while those more exposed to migration experience greater mixing. Otzi's genome, with its strong ties to early farmers and lack of later steppe ancestry, told the story of a group living apart, holding on to traditions that elsewhere had already shifted. This isolation had consequences not just for culture but for biology, potentially contributing to reduced genetic diversity. Such findings invited reflection on how isolation can preserve certain traits while also limiting adaptability. His DNA became a case study in the trade-offs of genetic seclusion, continuity on one hand, vulnerability on the other. Beyond the science, the revelations from Ötzi's DNA provoked questions about how societies use the past to construct identity in the present. The discovery that he had darker skin and Mediterranean features challenged long-standing assumptions about what ancient Europeans looked like. This disrupted simplistic narratives and reminded the world that human diversity is not static, but constantly evolving. His genome acted as a mirror, forcing modern societies to reconsider how they imagine their ancestors and, by extension, themselves. It was a reminder that history is not a neat story of continuity, but a dynamic, often surprising tapestry shaped by migration, adaptation, and change. If you liked this video, please give it a like and subscribe to our channel. Also, leave your comments below and tell us, what are your thoughts on scientists decoding Otzi's DNA? We want to hear from you. Thank you for watching and see you next time.